All right, so the session is recording, and this is good because we'll be hopefully able to address all your questions today. So we're going to start off as we normally do, and we're going to go over a few of the key slides and discuss them in a bit more detail. If you have any questions about them, feel free to raise your hand or write in the chat and we can discuss it further. And then I have some sample problems saved for the end of the session. And I believe that Lou went over the assignment instructions last week, so I won't be doing that. You should have also had a pretty clear explanation in your tutorials. But if you do have any questions about the assignments, we can also discuss that at the end of the session. Does that sound good to everyone? Are there any questions before we begin? Thumbs up if that sounds good. Okay, awesome. All right, so quantitative genetics. The idea here is that we know many phenotypic traits are influenced by the environment. So for example, they might be polygenic, meaning multiple genes control the particular trait, or multifactorial, in which case we have an effect from the genotype as well as the environment. And quantitative traits, or you may be more familiar with the term complex traits, it means the same thing. These can be explained on the basis of segregation of alleles at multiple loci. So multiple different regions of the genome are having some sort of effect on this complex trait. So those are quantitative traits. Now, when we talk about quantitative trait locus or loci, here what we're talking about is cases where we have individual phenotypic classes that are masked by the environment. So the difference here is that a quantitative trait locus is a region of DNA, right? So a locus that is associated with a particular phenotypic trait that varies in degree, which can be attributed to these different polygenic effects, right? So this includes two or more genes and their environment. So what do we do with this sort of information? So we can calculate a few different things, one of which is heritability. And heritability in its true term, that true term means it doesn't mean, but it is a measure of the genetic effects that are buried in a quantitative trait. So how much of the variation of a trait is attributable to genetic variation as opposed to environmental variation? And we can see that definition over here. So all of these different equations that I've got in boxes on this slide here, you need to be familiar with. So phenotype equals genotype plus environment. And then we have phenotypic variation equaling genetic variation plus environmental variation. And this equation here, this is going to be, and we'll see on the next slide, our broad sense heritability calculation. And these two terms can be further subdivided. So we can see our phenotypic variability here equating these two terms, whereas our genetic equates these three terms. So you can further subdivide this. That's what the connecting ideas are on these two, in these two different columns. And here we just have a summary of the different terms. So you can see that we have variation due to additive effects, due to dominance effects, and due to epistatic effects, which we're all familiar with. We've been learning this in the first half of the course. Okay, so let's dive a bit deeper here. Here we have our broad sense heritability, and this broad sense heritability is a measure that tells us how much of the phenotypic variation is ascribable or attributable to genetic variation. So what does that mean? We have genetic variation here, you can see, over phenotypic variation, right? So if our numerator here is large, that means that a large portion of that variation is attributable to genetic variation because this is over total phenotypic variation. If our numerator here is small and therefore we get a value that is closer to 0 for our broad sense heritability, that means that most of that variation is not attributed to genetics, right? Most of that variation instead would be attributable to environmental effects. So far, so good. Are there any questions before I move on? Okay, I don't see any questions. Again, feel free to use the chat if anything does come up as I'm explaining these different terms. Okay, so what broad sense heritability can tell us is that if it is high, that the phenotype of an individual is likely to be attributed to its genotype in that family. However, it does not tell us what phenotype an individual will have on the basis of its parents' phenotypes. So despite the fact that this broad sense heritability might be high, the precise phenotype cannot be predicted. And because the reason for that is what is occurring in these different families is very specific, very family specific. Now, narrow sense heritability is a little bit different. So again, we're still calculating this concept of heritability, but now we're specifically talking about additive genetic variation. And this is known as narrow sense heritability. So if we have a value closer to 1, then the phenotypic variation can be attributed to additive genetic variation. If we're closer to 0, then it could be attributed to other genetic and environmental effects. And just backtracking a little bit and taking a look at this slide. So remember, the other genetic effects include variation due to additive, to dominance, as well as epistatic effects. So when we're calculating narrow sense heritability, we're just focusing on this portion, whereas when we're talking about broad sense heritability, we're calculating this portion here. All right, so to further understand what narrow sense heritability is telling us, if it is high, then the phenotype of an individual is predictable based on the phenotype of its parent in that family. What it doesn't tell us is what is happening in other families. So these are the two distinctions in terms of understanding the concept of these different types of heritability that you need to be familiar with. Okay, so what do we do with quantitative trait loci? So there are 5 steps that you may be asked to carry out. You may be asked to carry out all 5 or just one of these 5 steps. So we're going to take a closer look at all of them. Yes, that would be further clarified in the question. So family would be, for example, like a pedigree. They would be close relatives, yeah. Okay, so let's take a broad overview of these different steps, and then we're going to take a closer look at each one, and we're going to dissect it a little bit more. So step 1 is to make an informative cross between individuals that are inbred relative to each other and which differ at the traits of interest. Then you would determine the frequency of distribution in the F2 generation. You could then use molecular markers to genotype the individuals and attempt to figure out which markers are co-segregating with the phenotypic trait that you're interested in. And then you can use a statistical method to determine if those markers are in fact co-segregating with the trait or not. And then you can plot the degree of association on a linkage map. So here we have another formula that you should be familiar with. So the odds ratio formula, which is the probability of linkage over the probability of no linkage. So here we've got our log of the odds ratio, or our LOD score. And that's how you'll see it plotted on the y-axis over here. And here you can see different loci. So they're given with these different letters on a particular chromosome. And then you can see that the log of this odds ratio is the probability of, or likelihood of, co-segregation of the trait and alleles at a locus. So it's a statistical estimate of whether these genetic loci are if the marker and the loci that you're interested in, so the one that is responsible for the phenotypic trait, are physically near enough to be inherited together. And what that tells you is that the association of that particular loci and the trait, if we get a LOD of 3 or more, is real. So there's a 99.9% chance that that association is real. So what you need to remember from this slide is the fact that if we have an LOD score of greater than 3, then we have a very, very high likelihood of a true association between this whatever genetic locus you're looking at and the trait of interest. Okay, so I know this is a little bit overwhelming, so we're going to take a closer look at each of these different steps. Is it just greater than 3 or greater than and equal to 3? Greater than or equal to 3? Yeah, great question. All right, so the step 1 was to make an informative cross. And here we can see that we have an inbred tomato plant with large tomatoes and then small tomatoes. And what this step is really emphasizing is the fact that they need to be inbred so that this parent has all of the same alleles, right? For whatever loci you might be interested in, it's going to have the same alleles. And this parent is going to have the same alleles on both chromosomes for whatever loci is responsible for this small size. So that's why they need to be inbred on this particular trait. Okay, and then once we carry out this cross, we get our F1 generation, we inbreed our F1 generations, we get our F2 generation. And in our F2 generation, we're going to get the full combination of genotypes and different phenotypes. Once that is done, you can determine the frequency of distribution in F2. So here, for example, you can see we've got 3 tomatoes that are very small, 4 that are medium, 3 that are very large. So you get this frequency of distribution. So you can see how these intermediate sized tomatoes are more frequent than either extreme. Step 3 is to then use molecular markers to genotype, and this is in quotations because it's not exactly genotyping in the strict sense, but it does help us pinpoint the loci that we might be looking for. So we're looking for markers that co-segregate with the trait. And here what we can see is, for example, if we didn't have this portion of the graph and we were to just look at our small tomatoes, we can see that all of them have this particular allele, this A2, whereas the intermediate size have one A1 and one A2, and then the large ones you can see have the A1 strictly. So that's what we're looking for when we're looking for molecular markers. And this can be done in a variety of ways. And later I have a sample problem which will work through something similar together. So you can see how this is done. And I see that there's a question. Are the different loci additive, or do they follow some other genetic relationship? In which context do you mean? Could you clarify? So in this case, they could be additive. We don't really know. we don't know yet. We would need to do a few more analyses. So they could be additive. There could also be a dominance relationship. There could be some epistasis occurring, and it could be two of those three or one of those three. It doesn't have to be all three. No problem. Okay. Okay, so then we have step 4, and here what we're doing is we're using a statistical method to determine if the markers are co-segregating. So here you can see a very clean relationship between the segregation of these different genotypes, so these different alleles or the markers that we're looking at, as well as the size of the fruit. So you can see all the small fruit, they have this particular genotype. All the medium-sized fruit have this one, and all of the large ones have this one. So this is a very nice, clean co-segregation and association between these two. And step 5 is then going to be to plot the degree of association. So the logarithm of the odds ratio, or the LOD score, on a linkage map. Again, you see this equation here. Make sure that you are familiar with this equation and that you're familiar with how to calculate the logarithm of the odds ratio. And here we've seen this chart before. I spent some time explaining it. The biggest takeaway from here is that once we have an LOD of 3 or more, we know that the likelihood of association due to chance is actually low. This should say low. Oh yes, so it's a low likelihood of association due to chance. There's a very high chance that the association between this marker and the phenotypic trait that we're interested in is real. So that's what that means. Okay, so that brings us to the end of all these summary slides. Before we go on to the practice problems, would anyone like me to clarify anything? I think when it comes to these concepts, they're pretty clear. It's mostly about applying this knowledge and being able to answer questions. Oh, great question. So when we're talking about additive, we might be talking about the effects among different genes. So an allele at one particular loci might, let's say, add 2 centimeters, whereas an allele at another loci might be adding 4. Does that make sense? And we'll see an example when we work through the questions as well. They can as well, but that would be a relationship between the two alleles of a single gene. And when we're talking about quantitative traits, we're talking about the relationships between different genes that contribute to a particular complex trait. And a genetic marker, that could be anything. So it could be a SNP, it could be, usually it's a SNP that we associate with a particular trait, but that doesn't mean that that SNP is even part of the gene that's involved with influencing the phenotypic characteristic. That SNP might just be very, very close to the gene that's responsible for that trait. So we call it a marker because further genetic tests would have to confirm if that SNP is responsible for the phenotypic effect, or if it's part of a gene that's responsible for the phenotypic effect. Is that clear? I'm not getting a thumbs up or follow-up questions. Yes, exactly. You've got it. So the marker doesn't necessarily cause the variation. You've got it. Okay. All right, so as we normally do, I'm going to display the first question, give you guys all a moment to participate and try and get the answer, and then we'll work through the solution together, and hopefully everything will make sense by the end of the session. Okay, so here's the first question, and I'm going to start the poll. Oh, there's no question. Here we go. Now we've got the first question up. And once I get around 50% of you participating, I will stop the timer and we'll take it up together. All right, so let's take a look at the different over how we would solve this question here. So some of you did get the correct answer. The biggest thing with this question is that what you would want to do initially is try and simplify it for yourself. So your first step, whenever faced with a long question stem like this, is to just take note of the most important information. So in this case, what we want to first note down is are these different alleles and the length that they add. So you would want to note that down as you're reading through it. And so here I've got A1 adds 3 centimeters, A2 adds 5, A3 adds 1. B, we're told, will add 2 centimeters. Unless it's B2, then it's going to add 0. And so what else we would want to do with this problem is to simplify it. So we're being told that we're crossing 2 hedgehogs with these genotypes, and we have genotypes for 2 different loci. So we've got the A and the B gene. And you can see at the B gene, at the B locus, we've got homozygous individuals that we're crossing. So our F1 is going to be heterozygous. So no matter what, the only combination in our F1 for the B locus is going to be a single B1 and a single B2. So because of this, we are always going to have plus 2 centimeters from the B locus in all of our F1 generation. So what you really need to focus on is just what is happening at this A gene. So what are the different combinations of A alleles that we might get? So using a Punnett square, you can note them out like this, and you get your different combinations of A alleles. And then because you noted all these different values down, you can quickly add them up and see that, with this combination, we would get 8 centimeters, 10 centimeters, but we're looking for tail lengths of 8 centimeters or less. And we know no matter what, we're going to have plus 2. So our A, our A has to be less than or equal to 6 centimeters. So both of these would be too long. So only these 2 options, so 2 out of the 4 would give us a length that is acceptable, which is 8 centimeters or less. So this is 50%. The answer is B. Thumbs up if that's clear. Let me know if you have any follow-up questions. And in the meantime, I'm going to address the questions that I see in the chat. Can I go over what a marker means again? Absolutely. So a marker is anything that you're using to sort of trace the location on the chromosome that might be segregating with the phenotypic trait of your interest. So a marker could be a gene. A marker could be a SNP. It is just something that you can identify, but it doesn't necessarily mean that that location is what is causing the phenotype that you're looking at. That's why we call it a marker. Does that make sense? Okay, awesome. And can everyone give me a thumbs up if the answer here makes sense? Let me know if you have any questions about it. Okay, awesome. Why add 2 centimeters? Great question. So we're crossing 2 hedgehogs with this allele at the B genotype and then this allele. So all of the F1 from this cross is going to be B1, B2 for sure. And we know that B1 adds 2 centimeters, B2 adds 0 centimeters. So no matter what, we're adding 2 centimeters from our B locus. Awesome. I'm glad you get it. So we always have this plus 2. So we're looking for A loci, or for A alleles, I should say, that are going to give us less than or equal to 6 centimeters from the A. All right, let's take a look at the next question. All right, and here we have the answer options. So I'm going to restart the poll, and once you have the answer, feel free to vote, and then we'll take it up together. All right, so let's take a look. This question is telling us that a researcher is studying the heritability of birth weight in humans, and that this trait shows continuous variation. In one family, different variances for the F3 group of siblings were calculated as follows. And here we can see all the different values, and we're being asked which of the following statements is correct. So what conclusions can we make based on the information that they're presenting us with? So initially, what we can calculate is our phenotypic variation, because they give us all these different values here. And we know that, that the variation, the genotypic variation and the environmental variation, when added together, will give us our VP. And we can calculate that because we have that information. So we get a value of 100. Then we can also calculate our broad sense heritability, which, as we've discussed today, is VG over VP, which we can further subdivide into all of these different genetic effects over our VP, which we know we calculated is 100. And we get a value of 1 when we do that. Then we can also calculate our narrow sense heritability. And what's really interesting is that the only genetic effect we're getting is from this additive variation. So that is going to give us the same value. We get a value of 1. So based on those calculations, we can determine that B is the correct answer. But since we have some time, let's take a look at the other answer options and see why they are incorrect. So here, A is telling us that our broad sense heritability is equal to 0. We've already calculated this as not true, right? We know that our broad sense is 1. C, neither broad nor narrow sense heritability can be calculated. Again, not true. We're not dividing by 0, right? Our VP is 100, so this statement is not true. And then our narrow sense heritability is equal to 0. Again, not true. We've calculated it as equaling to 1. Thumbs up if that's clear. Let me know if you have any follow-up questions. Okay, awesome. Let's take a look at the next question. Oh, I see that there's a question. Max is always 1. Yes, you've got it. 1 is the absolute maximum. Okay, so I'm going to relaunch the poll. And then we'll go over it together. All right, so let's have a look at this question here together. We're told that a researcher at Sick Kids is studying the heritability of birth weight in newborn babies, which is a phenotypic trait that is continuous. And for babies born in Toronto, we see the following phenotypic variances in birth weight, and they're attributed to these different effects. And the question is asking us what is true concerning the birth weight of babies born in Toronto. So sort of like what we had before, we're going to have to see what sort of information we can extract based on the information that's being provided here. So in this case, we can just work through the different answer options and see how they are, whether they are true or whether they are false. So A, most of the phenotypic variation in birth weight in this population is due to environmental effects. We can see that without even doing any math, right, our genotypic variance is higher than the environmental one. So A is incorrect. These three are going to add up to more than the environmental effects. And these three give us our VG. B, for this population, broad sense heritability is lower than narrow sense heritability. So in this case, we're going to have to do a bit of a calculation. So let's see what that would look like. Here we have broad sense heritability. And again, we're familiar with this equation here, and we know that we can further subdivide this into these three. So we can calculate it as 55 over 100, or in this case, we get 0.11. Narrow sense heritability, we can calculate as just the additive genetic effects. So just this 40 over here, and we get a value of 0.4. So is broad sense heritability lower than narrow sense heritability? Can anyone tell me? It's higher. You've got it. We can eliminate B. Now, C, we've calculated. We know this is true. This is the correct answer, but let's take a look at D and see why it's not true. Most of the phenotypic variation in birth weight attributable to genetic effects is due to these dominance effects. And again, that is not true. We know that most of it is due to these additive genetic effects. All right, any questions about this? Thumbs up if everything's clear so far. Fantastic, look at you guys go. Yes, I can explain why it's not A. So phenotypic variation would, due to environmental effects. Yes, you're correct about that, right? But remember, these three add to genetic effects, right? So our VG is equal to our additive effects plus our dominance effects, as well as our epistatic effects. So our VG is actually equal to 55. And when we compare, what this statement is saying is that the phenotypic variation is due to environmental, most of the phenotypic variation is due to environmental, as opposed, it should say, to genetic. Because we wouldn't be comparing our environmental directly to just, if it said, for example, as opposed to additive genetic effects, you could say this is true. But I could see where the confusion would stem from, because this isn't being specified. But whenever you're comparing environmental or phenotypic variation, you are assuming that we're talking about the genetic effects and environmental effects, not the subcategories of these different genetic effects. Yeah, awesome. Okay, so for all cases, broad sense will always be greater than or equal to narrow sense. You've got it. Yeah, it can't be less, because it itself encompasses a portion of the narrow sense heritability. And that kind of makes sense, right? Because broad sense, think about, broadly covering the genetic effects, whereas narrow sense is focusing in narrowly, is that a word? On the additive genetic effects. Does that make sense? So broad sense will always be greater or equal to. No problem. That's a great observation to make. It will be helpful. For example, if you're stuck on a question and you can't figure it out, at least you'll know that and you can help yourself by eliminating a few things. Okay, so let's take a look at the next question here. What's our next question? There we go. And I see there's another question in the chat. Will genetic variation be only composed of these three subcategories? Yes, you've got it. So our genetic variation, if we go back to our earlier slides over here, so our genetic variation, it's going to always be composed of these three. Sometimes, like I said before, some of these might equal to 0, right? And in that case, it might just be one of the three that contribute to it. But it will always be at max these three. All right, awesome. Okay, let's go back to this question here. So this is purely a memory question, a memory style question. So either you know the answer or you don't. I'm going to give you a moment to vote. And if you don't know the answer, don't worry about it. Just take your best guess and we'll take a look at the table after. So this was a question in the past, and it would be helpful to know the general trends and the outliers, but I wouldn't worry too much about memorizing all the individual values. But you might want to confirm that with the prof. Okay, so I got a few votes in. I'm going to end the poll. And as some of you have suggested, this does come from this table over here, this table that was shown in your last, not today's class, but the one before. And yes, exactly. So I don't think I would worry too much about memorizing the exact percentages here, but it is helpful to know the lowest or the highest ones. Okay, and most of you did get the right answer. So litter size over here is the correct answer. Any questions about this? Again, this is very much just a knowledge-based question. Not much I can explain here. Okay, so let's take a look at another sample question. Here we are being introduced to sort of a different way of thinking, and I'm going to create a poll. Again, this is a relatively quick question to answer, so take your best guess and then we'll discuss it together. Okay, so we have a 50-50 split with this one. The correct answer is D. The reason being that most cancers are caused by environmental factors. So we assume that her risk of developing cancer is going to change because the reason that their family was commonly diagnosed with this cancer was because of their environment. And now that she's emigrated, it's no longer the case. So D is the correct answer here. All right, let's have a look at this question over here. And this is similar to something that I brought up earlier. Oh, I see there's a question about the earlier one. So before we move on to this, let's talk about this a bit more. So the reason is because cancer in itself is most cancers are caused by environmental factors. There's a very strong environmental component to them. So the fact that she is changing her environment, because the question sum tells us that, we can assume that the risk of developing this cancer is going to change as well. And if, for example, let's say you were unsure, you can also answer this question based on process of elimination, right? So A stays the same. She has a 50% chance of inheriting stomach cancer. Nothing suggests this, right? We're not given any information. We know that cancer is a complex trait, right? That is something that that you should know going into the exam, into the final, that traits like cancer are complex traits. They are complex diseases caused by a combination of genetic and environmental factors, right? Let's say you didn't even know which was larger than the other. So there's nothing to suggest to us that it's going to be a 50% chance of inheriting stomach cancer. There's nothing in the question sum that can help us make that sort of calculation or make that sort of inference. So for that reason, A is not a good option, right? Change, cancer is more likely to be detected early in Canada. Again, nothing in the question stem is suggesting this. I don't think that it would be easier to detect in Canada. It might just be just as easily detected. So B, incorrect. And then stays the same. She has a 25% chance of inheriting stomach cancer for the same reason as we saw in A. We have no information given to us in this question stem to be able to make that assumption. So D, just through process of elimination, is the best option, but it is also in fact the correct answer because cancers are caused by environmental factors. Okay, glad I could clear that up. So let's take a look at the next question. Genotype does come into play for cancer, for some cancers, right? So if we think about breast cancer, for example, but not all cancers. And even when it does, it's a much lower, it has a much lower effect than the environment would. No problem. Okay, so question 14, I believe this is the poll I've relaunched for it. Yes, okay, I see people voting. So this is a poll for question 14. I'll give you a few moments and then we will discuss the answer. All right, so there was a close split between two different answers, either C or D. And let's take a look at the difference between those two, because what really differs is whether this cross is occurring between different inbred lines or the same inbred line. And if we had the same inbred line, can anyone tell me, would those two different species have the exact same genotypes or no? Yes, exactly. They would. So if they were the exact same inbred line, they would be homozygous at the same loci. So even if we had a large pod rat and a small pod rat, and they were homozygous everywhere, or I should say inbred everywhere, and homozygous at those loci, what that would actually tell us is that this is purely environmental, right? Like no matter, they have the same genotype, but different phenotypes. So in order to, because we're being told it's a quantitative trait, we know that there are different genes responsible for this trait. We need to take a look at a large pod rat and a small pod rat that come from different inbred lines. If they come, this statement doesn't make sense. They wouldn't be able to come from the same inbred line, but they do both have to be inbred. And where we see an example of this illustrated is here, where we take this large tomato and the small tomato, and you can see that parent one is homozygous for all its loci, and parent two is homozygous for all its loci, but together they are not the same inbred line. They are just two separate, two different inbred lines. Thumbs up if that's clear. What about B? Oh, so that wouldn't be what you would do initially, right? First, what you want to do is you would want to take a large rat and then a small pod rat and cross them in order to then generate your F1, which would be the medium pod rats. Because in that case, that's where you would get the one chromosome from each parent, one allele from each parent, and then you would get the mix that would be informative for you. So this would be most useful. C would be most useful to begin this experiment. If you just started with the two medium pod rats, you wouldn't know if, for example, you're capturing this over here, or if you're capturing this, and this would be less informative to start with. So it's like if parent one is AA and parent two is AA. Exactly, except you have to remember that these are quantitative traits, right? So we're talking about AA and BB and CC. Yeah, but you've got it. That's the right idea. All right, so let's take a look at the next question. Oh, I see that there is a follow-up. So if D was true, it would mean that variability is environmental, right? Yes, exactly. So if, for example, this question stem, how could I reword it? I mean, what would end up happening is that that would somehow be incorporated into the question stem, right? Saying that from the exact same inbred line, meaning they have the exact same genotype, you've got this and this. What does this tell you about the phenotypic variation? What must have the larger effect? Is it the genotypic effects or the environmental effects? And that's actually a great question to ask, because you might be asked, for example, how would you design an experiment to test solely for the environmental effects, right? And the way that you could do that is by keeping the genotypic effects the same, aka using the exact same inbred lines, but varying the environment. Does that make sense? Thumbs up if that makes sense. Or am I just rambling now and losing everyone? I don't see any thumbs up. Okay, so phenotype only depends on environmental effects. No, oh, for D. Yes, yes. If that were true, then yes. I mean, you would have to carry out an experiment to confirm that, right? So you would need to use the same inbred line and, for example, grow them, one in a cage and one in a field, and see, oh, the one that grows in a field has larger paws because the environment, affects that phenotype. But yeah, you're following. You've got it. Okay, so let's take a look at the next question. Thumbs up if this is clear. Okay, awesome. And C is our correct answer. Okay, so as I promised, I said that we would do some of this mapping, right? Some of this quantitative trait loci mapping. So I'm going to give you guys a moment to take a look at this question, relaunching the poll. Please vote, and once we have a few votes in, we'll discuss it together. Okay, so we've got a kind of even split between three options, gene C, D, and A. So let's talk about how we would tackle this sort of question. So the first thing you want to do is identify the high versus low groups. So just roughly taking a look here, we can see that one is 50, one is 51, and one is 49. Those seem like pretty high values, so we can group those together. And let's take a look at the other group, which would be probably 30, 33, 29, and 28. So here I've, well, I've given the answer, but let's take a look at the groups, right? So here is our, in green, we can see our large or higher groups. And in red, we can see the lower ones. And what you want to do is you want to, let's just focus in on one. So let's focus in on the green. What you want to do is see which of these segments is consistent across these three. So the white will be inherited from one parent, while the black color indicates that that one is inherited from a different parental line. So taking a look at 53, we can see that here our A is white, but when we take a look at our, we take a look at our 51, A is white as well, and 49, A is white as well. So this could be an option. Let's take a look at B. B here, our B is dark, and our B here is light. So we know that B is not an option. C, here we have a dark color, here we have a dark color, and here we have a dark color. So C is another option. So we've run into a little bit of a problem. Let's take a look at D. D is the same situation, right? Dark, dark, dark. So now how do we then further identify which one of those three it is? So let's take a look at our red group. And our red group, if we take a look at region A, we've got a white segment, and then we have a black one. If this were the region responsible for the, I don't even remember what this was, average estradiol levels, if this was the region responsible for that, we would expect 30 and 33 to be coming from the same parent. In addition to that, we would expect it to be coming from the other parent, not from the same one that is causing this high percentage, high concentration. So for that reason, A is incorrect. We've already crossed out B. We know that one doesn't work. Let's take a look at C and see if C aligns. So C, yes, we can see that this is the same segment in this whole group coming from a single parent, and that parent is different than the higher group. Let's take a look at D. And D, we run into the same issue, right? Here we have the segment coming from the same parent, yet they're part of two different groups, and it's not consistent among these groups, among the low group values. So the correct answer here is C, as I gave away. But is this clear to everyone? Does everyone see how to solve this? Thumbs up if it's clear. Are there any follow-up questions? More practice. I think this is the only practice I have here, but we could take a look. At the end of the session, I can see if I can pull up some more practice. For sure. And I see that there was a question I think I missed. So oh, I see. No, so because they're inbred, they're going to be at all the loci. No, okay, I see what you're saying. No, they don't have to be homozygous dominant for all the loci. They could be a mix of homozygous dominant or homozygous recessive. So yes, that is definitely possible and likely going to happen. What do I mean coming from different parents? I mean that, so if we were to backtrack a little bit, let's take a look here. So kind of like how we were looking here, but here we use the colors. Oh, what is happening? There we go. Here we use the colors green and pink to indicate which parents is coming from. Here you can see that this segment is coming from parent 2, while this segment is coming from parent 1. Right? So the colors are just indicating the fact that they're coming from different parents. It doesn't actually matter which parent it's coming from for the purpose of this mapping right now, because we can determine that based on the segregation among the different groups. Yes. Yeah, so it would be the F2 progeny that we're looking at for these maps. Right, exactly. You've got it. So in our case, we could say that the white is coming from parent 1 and the black is coming from parent 2. Yeah. Is it only divided into two groups? I'm not sure I understand that question. What do you mean by only divided for the practice questions? I'm still having trouble following, sorry. Is what divided into two groups? Oh, you mean this? Yes, yes. So you would take, you would look at the extremes. Yeah. So in the F2 generation, that's where you're going to get your distribution, your full frequency of, or your full distribution of phenotypes, and you would specifically analyze the small and the large, right here, or here the low and the high, in order to get those loci for this form of mapping. So there could be three groups, but you would look at the low and the high. They would be more informative. Is that clear? Okay, awesome. All right, so we have, what time is it? We still have two more questions. So before I go back and we practice more of this, I'll go through those last two questions. They're they're not too long, so we will have time to still go over another example. All right, here we go. I will relaunch the poll and we'll regroup. All right, so let's have a look at this answer here, or this question before we get to the answer. So here we have a test cross with a plant that is heterozygous, and we're told that from the cross, 6 of the progeny had a recombinant genotype and 9 were parental. So we're being asked, what is the specific LOD score for this specific marker and the fruit size gene? So how do we go about solving this? So we know that our LOD score is the log of our odds ratio. We remember this formula that I mentioned, the probability of linkage over the probability of no linkage is our odds ratio. And what we're told is that we've got 6 recombinants from our test cross and 9 parentals. So from our total progeny, we can calculate our recombination frequency here. Our recombination frequency is going to be 6/15. We remember this from our earlier classes. And our parental frequency is 9/15. So we've got our 2 frequencies here. Now, bringing us back to this, in order to calculate our odds ratio, we've got to get the probability of linkage over the probability of no linkage. So our probability of linkage can be calculated by the recombination frequency to the power of the number of recombinants multiplied by the recombination frequency, this should be RF, the parental recombination frequency. Oh, sorry. Yes, okay. Let's not put an R there. So it should be the parental frequency, which is PF is correct. So the parental frequency here, let me write these in because it even confused me for a second. So PF and then RF. Here we go. Of our parents or of the parents. And then all of this is going to be the over the probability of no linkage, which here probability of no linkage is always going to be a 50% chance, so 0.5 to the power of the total number of progeny, which we've calculated as 15. So here we're just plugging those values in and we get our odds ratio. Now, be careful because we're being asked about the LOD score. So some of you selected D, which is the correct odds ratio, but we're looking for the LOD score, which is the log of the odds ratio. So the correct answer is B, 0.13. So the probability of no linkage is always 0.5. And then you have the total number of progeny from the question stem. We've got the 6 recombinants and 9 parentals. Okay, so to sort of extend this question, I see there's a few comments in the chat. I'll get to them in a sec, but I'll pose this question to all of you right now. So with this inference, with this statement, could you conclude that the association is real? No, exactly. You guys have got it. You're all experts. Okay, let me see what questions I have here in the chat box. Okay, for the numerator, why don't we consider the fact that the order could be different, like in section 1 when we had so shouldn't the numerator be multiplied by 15, choose 6? Good question. So in this case, we don't have, we're not given that sort of information. We're not given anything about the birth order. We're just told the number of recombinants and the number of offspring that have the parental genotype. So in this case, you wouldn't need to do that. We could also multiply the numerator with the same number, so it will cancel out. I think I'm not following that statement. Give me a moment. Multiply, if we are taking order into account, we could multiply the numerator with the same number. I don't think you're misunderstanding it. I think you might be overthinking it, because I'm not following why you're multiplying the numerator or with what you're multiplying it. The denominator with 15. Yeah, you could. I think this way. Yeah, exactly. So this way you're simplifying that calculation. Okay, yeah. So it's not necessary for that reason. This would be the efficient way of solving this. Okay. No, that's all right. That's all right. I'm glad we were able to figure it out. Okay, so are there any questions about this before I move on? So we have just one more question, and this has to do with the LOD score again, and it's just a knowledge-based question. So I'll give you guys a moment to answer that, and I'll relaunch the poll. And then we will take a look at some more mapping. Okay, so most of you got the correct, or actually all of you got the correct answer here. So the correct answer is in fact D, and we can discuss why that is in a bit more detail. So D is the correct answer, and here we've got a few equations that we are, or just one equation, I guess, that we're familiar with now. We used it in our earlier question. And here we can see the graph illustrating what this association means, right? So A, it is equal to the probability of no linkage divided by the probability of linkage. This is not true. It's actually the opposite. It's the probability of linkage divided by no linkage. An LOD score of 3 means there is a 1 in 3 chance in favor of there being a QTL. No, that's not what it means. LOD of 3 means this, as we discussed. So in fact, it's a much higher chance that the association is real. LOD scores of above 1 are generally considered good evidence for a QTL. Again, nope, this is not true. We are looking for the value of 3. So D is the correct answer here. None of the above. Thumbs up if that's clear. I think it's clear because most of you got the right answer. Okay, awesome. All right, so that brings us to the end of this session. Before I stop the recording and we get to the question period and I do the extra mapping, I do want to show this slide as a reminder that you all have an assignment due November 14th. It's worth 10%. You have to submit a, I see that there's a follow-up question for the earlier question. So let me just address it. For B, it should say 99.9 instead of 1 and 3. Yes, that would be correct. You've got it. Okay. So there's 100% late penalty, right? So what does that mean? That means that the second it's late, you're getting 0. So make sure you hand it in, make sure you do it ahead of time so that if something happens last minute, you don't have to, ask for an extension if that is even going to be granted. So just make sure you get it done, start working on it. It involves a 500-word, one-page essay about the genetics of a single gene disorder. You can find a list of them mentioned in this article that is linked on your Quarkus page. And the second part, and don't forget about this part because students sometimes forget, is to create a pedigree, right? So this can be done by hand. If you do choose to do it by hand, make it nice and neat. But the pedigree is going to be a disease or condition that it's going to be, a pedigree for the disease that you've chosen to write about in part A. And it's made up, right? So you can be creative, still be realistic, but it doesn't have to follow a particular pedigree that you know of. Okay, so are there any questions before we move on? Is there a strict word limit? Yes. So there is a strict word limit, 500 words, right? With 10% give or take. It is strict, do not go over it because you don't want to lose marks for that. So if you find that you're going over it, try and be more concise in your writing. That's a sign usually that you've added a lot of content, but you need to sort of word it more concisely. The limit does not include references. No, it does not include references. And the title would be part of that, 50-word give or take. So your title, it shouldn't be over 50 words anyway. No problem. Oh, in-text citations. Great question. So can anyone tell me what kind of citations are we using for this assignment? What kind of style are we following? Yes. And what does nature look like? Does anyone know? It's not footnotes. It's not footnotes, but it's just a number. It's just the word escapes me right now. The number that's really small and at the top. What is that word? Superscript. There we go. Thank you. So it's just a superscript that you're going to be adding, and make sure you do that. Don't do MLA, don't do APA. We're doing nature, right? So make sure you follow that style. So it won't affect your word count. Okay, awesome. So I'm going to stop the recording now. That brings us to the end of